Cisco and no. Any other questions? Alright, I'll give you topics for every single question right now. You ready?
Yes, okay.
Get this on camera.
Okay.
Record this.
Number one is about chromosomes. That's where you find them. Need to know the difference between like genotype and phenotype, right? Hey, how's it going?
What up?
Continue class, please.
This is helping me study. Don't worry about it. Act natural.
There's no dihybrid crosses, but there are some simple crosses of simple Punnett square.
Why are you looking at the camera?
I thought
It's
you
not
got
natural.
enough experience with dihybrid cross on the activities.
Her head's in the way.
You're so ready for it.
This is natural.
Let's see, you need to know what carries the information from the gene in the nucleus to the ribosome.
Cytoplasm.
No, it's not.
I'll give you an answer. It's not cytoplasm.
Dang it!
How does the
I think
gene,
it's
the
translation.
information that's on the chromosome,
No, it's
get to the ribosome?
Transcription. Transcription?
Does DNA, yeah, exactly. Does DNA leave the nucleus?
No.
not by itself.
RNA does.
Which RNA? There's two
mRNA.
types.
mRNA.
The messenger, right?
mRNA.
Is that what it stands for?
And what was the question?
Okay, so what goes into the nucleus and gets out the DNA? That's the question.
What?
And sends it to the ribosomes.
It's the mRNA because it's the messenger RNA.
The
I'm gonna need you to make a copy of this.
How many chromosomes
That's horrible.
are in humans?
46.
23
How many pairs that is?
or 43.
23 pairs, 46 chromosomes. That was for you, Brianna.
23 pairs.
I'm gonna like make a video of myself. I'm gonna like dance and it's gonna
Don't do that.
No, all the vocab where we
next
talked
slumber
about
party.
what chromosomes
Alright.
were and when there's two of them, like what
that?
it's called. and
Called a
when
sister
they're-
chromatid.
When there's a set of them.
Oh, that's a
for
sister
each
chromosome.
parent. It's what?
Sister chromosome. And then chromatid.
What are the ones where each parent contributes to their like chromosomes?
Sister. No?
Hey, you guys! I'm trying to study
Be
here.
quiet, really?
I just gave her an answer.
Exactly, because people
It
are
wasn't to a
freaking
specific
talking.
question.
Good lord. Turn shut up.
Where's Daniel when you need him?
I
told him that was all right.
You need to be able to compare and contrast plant and animal cells, so there's some questions about which organelles are found in each or not found in each.
I do too. That's what I'm waiting for.
What's the 8th page?
Slowly like sliding down.
He's still on the first page.
Sir, can I go to the restroom?
If you can,
Tanner.
let's see.
What if I die?
Took a picture while I was here. I took a picture.
The activity that we did yesterday was an example of
You had dihybrid cross?
Oh,
sirloin
book. I don't even know where this is from.
Or wolf.
Oh, Jack in the Box. Dr. Pepper.
no. That's not the answer.
What's an example then?
So, Mr. Bergman.
The jobs of different organelles. Pretty easy stuff.
Oh, okay.
Started.
So the mRNA, what is it kind of RNA?
Know your phases of mitosis.
I know mRNA, that's what goes
only
after.
kind you need to know.
You want to recognize phases of mitosis.
Base of mitosis.
And meiosis?
Know about
Prophase,
haploid and
anaphase.
diploid and how many daughter cells for mitosis and meiosis. You guys did that on your compare and contrasts. Some. Yes.
Do we have? Okay. Do we have to describe what each phase does, like in mitosis and meiosis, or do we just have to identify the phases?
Both.
What has
There's some questions that have a picture and you have to identify what phase it is.
So,
Meiosis and mitosis.
oh, okay.
you say which one is mitosis and which one is meiosis, or we will say identify which one and if it was.
Oh, I tell you that it's meiosis.
Oh, looks really cool. Okay. Okay, so let's choose.
Let's see.
Whose pen is this?
Yeah, we're talking
I don't know.
another cross.
So there's no dihybrid crosses? Just a Punnett square? Okay.
And the jobs of cell parts. Let's see. What causes a baby to be male or female? Like what determines that? Other terms: homozygous, heterozygous, phenotype, genotype, and this really cool 2 words that I made up.
Is it paragram?
We actually made that one.
No, you made it up first.
Why does Hannah get to see the test? I want to see the test.
Zygos.
I can't.
Zygos is not the right answer.
Oh, he moved too fast. Wait, ready? Got a good.
Heterozygous is not the answer.
I'm Tiga.
You mean Tiga?
Tiga.
Did anybody happen to find the difference between RNA and DNA?
What? RNA and DNA?
RNA is a copy that turns into protein.
Deoxyribonucleic acid is DNA.
Yes. RNA
RNA
is
is the copy of
single-stranded,
DNA.
DNA is double-stranded. RNA can leave the nucleus, DNA cannot leave the nucleus.
Wait, RNA is.
Yeah.
RNA has uracil instead of thymine.
And oh no.
RNA has uracil instead of thymine.
What's your
RNA has uracil instead of thymine.
That's the answer.
Oh, I'm recording this. Does anyone want to come to my house and watch this?
What was the what?
Okay, so
You're still not thymine?
Another purpose of mitosis and meiosis.
Purpose?
cell division.
You know the molecules involved in a strand of DNA? What parts make up the ladder part, like the rungs of the ladder and the sides of the ladder? Yes?
Is most of this going to be multiple choice?
Most of it.
Okay. So
41 out of 40.
Taylor, what does DNA stand for?
Deoxyribonucleic acid.
And RNA? Ribonucleic acid?
Yeah.
Okay, just making sure.
Transcription and translation. What is purpose?
Ha, Tanner. Spacing Berman now. You can get up, loser.
Oh, remember the people that were involved in discovering the shape of DNA?
Or it's tied this way.
Your
I don't know,
thighs
because
is
people
high.
keep talking.
Don't invite people in. Never do that.
And Taryn got white out on my leg.
Ready? You want me to do
It's a
it
white
nicely?
substance on my leg. Not with a
On the outside?
Anti-parallel and
You tried to do
DNA
on the side.
structure.
I wouldn't let her.
Who knows anti-parallel DNA structure?
Anti-parallel?
Not parallel.
Is that like perpendicular? Just kidding.
Yeah, that
When
would be
it's all
nice.
screwed up, right?
It's gonna be
When it's not parallel.
No,
When they're
what happened,
jumbled.
it was Jamie that asked me a question when you guys were putting these together.
If cancer, if like
What you
the,
asked Jamie?
is that
was
how
it about the
When you were cutting them
twins?
out?
Oh,
That was me, wasn't it? When I asked why are some of them have to be upside down? Why do they have to be upside down?
Yeah.
Yeah. For
Because
me.
they're anti-parallel.
oh.
Okay, I got that.
No.
You guys notice when we tape these together,
But some of them are upside down.
that one side is upside down and one side is right side up, so they're actually facing like that?
Yeah, I knew that.
That's called anti-parallel.
Oh,
They
brilliant.
fit together. Oh,
And that pen.
Oh, yeah.
So they have
They fit together like bubbles in suds. I know, I just made that up. Right? Like bubbles in suds.
come together.
We should make a news broadcast.
Know who Jacob and Fernand were?
Jacob and who? How do you say?
Oh, there's my pimple. Are you really doing this, Pam? Pam, why are you doing this? It's cool. You just want to zoom in on your pimple?
My pimple on my face.
Oh.
I don't know because I can't hear.
Okay, it is not.
Can you guys be quiet, please?
Look at my pen.
Oh, she broke it.
I did not.
Hi.
Alright, and then
What
there's
fell asleep?
2 essay questions.
Oh, shh.
You got that? Did you zoom in on that?
Yeah, I got it. I'm gonna like
I'll tell you what they are.
filter this later so I can see it better. You guys, shut. You were saying, Mr. Bergman?
Basically the questions are the essential questions. The guiding questions here. These are basically the essay questions right here.
On this one, I did a little bit more background information, but thinking about the movie Gattaca and how DNA is used, it can be used to your advantage, but it can also be used to as a disadvantage. Okay? Like if you had a crime against you and you wanted to have that crime solved, you would probably be like, yeah man, we need to use some DNA, get this dude up in here, let's get him prosecuted, right? But if you were innocent and your DNA happened to be there at the crime scene, then you might feel differently about them kind of invading your privacy and then, you know,
We got you, Tanner.
for a crime that you had nothing to do with, and then they had no other proof than they found your
Too
DNA
bad.
at the place. Okay, that's kind of what the
You
first
have
one's
your
about.
whole
The second
model
one is
lesson thing
about
going on.
this
You
question
should
right
be on
here.
camera.
Okay, and that's what.
Woo!